Introduction to Host Defenses. In the last lecture, you learned about how organisms invade our bodies and how they start the infectious process. In this lecture, you'll learn about how the human body fights back against foreign invaders with our immune system. Immunity is defined as the capability of removing and/or killing foreign substances, also called antigens. Our immune system is grouped into innate immunity and adaptive immunity. The innate immune system is fully functional at birth without any previous exposure to antigens, so it does not need to be activated because the components of the innate immune system are all set and ready to protect us. Adaptive immunity is initiated and amplified by exposure to foreign substances, so it's activated when it comes in contact with an antigen. A healthy immune system must be able to surveillance the area like guards. There are cells that are on the lookout for foreign invaders. These cells must also be able to recognize these antigens and distinguish between foreign substances and our own body cells. We don't want our immune system killing our own cells. That would be an autoimmune disorder. The third role of the immune system is destruction. It's not enough to surveillance the area and recognize the antigens, but our immune system must also be able to get rid of them, preferably before they cause infection. The immune response to antigens is like a war battle or a game of chess. The first and second lines of defense are part of the innate immune response, always active and ready to go. The weapons in the first line of defense are like the pawns in a chess game. They're effective, but not as powerful as the second line of defense. If organisms get past the first and second lines of defense, they are faced with the hardest hitter, the queen, the adaptive immune response, also known as the third line of defense. The third line of defense must be activated, and when the infection is over, it remembers the antigen in case it tries to attack the body again. The first line of defense includes physical and chemical barriers. You should be familiar with all of these defenses. Chemicals are found all over the body. These chemicals can be enzymes as well as acids and bases to change the pH. Like in the stomach, the pH is very acidic, pH of about 2. When the food travels from the stomach to the small intestine, the pH changes abruptly and becomes more neutral. The low pH in the stomach kills many organisms, and then the abrupt change in pH as the food enters the small intestines also kills organisms. Lysozyme is an enzyme in tears and other body secretions that disrupt the bacterial cell membrane. Physical barriers of the innate immune system physically prevent infectious agents from attaching or entering the body. Intact skin is a physical barrier. It also contains fatty acids to fight pathogens that may try to adhere, and the skin is covered in normal microbiota, as well as our intestines, that physically play a role in preventing adherence of pathogens. In the nasopharynx and the trachea, we have cells that have cilia, little hair-like structures that trap foreign invaders. The flushing of the urinary tract serves to wash out any pathogens that try to enter those places that are open to the outside environment. The mucociliary escalator of the trachea consists of goblet cells that line the mucous membrane of the trachea and ciliated epithelial cells. The mucus traps debris and other junk that we inhale, and the cilia move those trapped substances up and out of the respiratory tract so they do not continue to travel deeper into the lungs and cause infection. Our innate immune response extends to our genetic makeup and offers resistance against organisms that cause infection in other species. When pathogens enter our body, they travel to the specific tissue that they normally would infect. The cells that make up that tissue have receptors that the pathogens will bind in order to enter those cells and cause infection. But if those receptors are not there, the pathogen cannot infect the cells. So this is why pathogenic organisms that infect certain animals can't infect humans, because our cells do not have the receptors for the pathogens to bind. Sometimes pathogens undergo some genetic mutations so that they can recognize human cells and attack them too. These types of infections are called zoonoses, and they can be transmitted from animals to humans. An example of this type of zoonotic infection is the bird flu. The second line of defense is also part of the innate immune system, but the defenses are a little bit stronger. Let's look at the different types of blood cells. This is the hematopoiesis flow chart that shows how the formed elements of blood, like the blood cells and the platelets, how they originate. They all come from this multipotential hematopoietic stem cell. This stem cell has the ability to differentiate into all types of cells that are found in our blood, the red blood cells, the white blood cells, and the platelets. The stem cell differentiates into a common myeloid progenitor and a common lymphoid progenitor. The common myeloid progenitor eventually becomes several types of cells. The megakaryocyte, where small fragments of this cell break off and form thrombocytes, also known as platelets, that are important in forming clots. The erythrocyte is the red blood cell, important for carrying oxygen throughout the body. The mast cell is important in allergic reactions. The myeloblast can further differentiate into white blood cells, such as the basophil, that is also involved in allergic responses, the neutrophil that plays a big role in phagocytosis, the eosinophil that's involved in parasitic infections, and the monocyte that differentiates into the macrophage or dendritic cell. Within the blood vessels or the circulatory system, the monocytes are just that. But when the monocytes enter into the tissues, they differentiate into macrophages or dendritic cells. The common lymphoid progenitor cell line can differentiate into natural killer cells as well as small lymphocytes, which further differentiate into T lymphocytes and B lymphocytes, otherwise known as T cells and B cells. The B cells can further differentiate into plasma cells, and we'll go into more detail about these differentiation of B cells into plasma cells as we go through the third line of defense. All of the white blood cells are important in the immune response. all that are enclosed in the red box, but we won't discuss them all in this course. For the pathogen, the human body is the host, and that pathogen is trying to infect our host cells. Our immune system kills our own host cells when something is wrong with them. For example, if it turns into a tumor cell or if the host cell is infected by a virus. Recall, one job of the immune system is to recognize host cells from pathogenic cells. All host cells that have a nucleus have something called MHC class I molecules on the outside of their surface. This is how immune cells can tell the difference between our own cells and pathogenic cells, because pathogens don't have these MHC molecules. In the top figure, we see a normal, healthy cell. This cell comes in contact with a natural killer cell, or an NK cell. The NK cell has several receptors that bind to the ligands on the healthy cell, including the ligand for the MHC class I protein. When everything is intact and normal on the healthy host cell, the natural killer cell detaches and continues to circulate through the body. When the natural killer cell comes in contact with a host cell that does not have an MHC class I molecule, like on the bottom, then there's a problem. For example, tumor cells and virus-infected cells typically lose their MHC class I molecule. So when it comes in contact with the natural killer cell, the only ligand that remains is the activating ligand, shown here. In the figure above, the attachment of the natural killer cell receptors to the MHC class I molecule prevents activation of the natural killer cell. But at the bottom, this tumor cell is missing the MHC class I molecule. So the interaction between the receptor, the activating receptor, and the activating ligand causes activation of the natural killer cell. When the natural killer cell binds the activating ligand, it signals to the natural killer cell that there's a problem with the cell and that cell needs to be destroyed. The natural killer cell then releases chemicals to degrade this abnormal or tumor cell. Recall that organisms have a temperature range in which they can grow, and above that temperature they will die, mostly because the proteins inside of the cell will also denature above that temperature, and without proteins the cell dies. The presence of microbes in the body causes it to increase in temperature. We get a fever, and that fever increases our body temperature above the normal range of microbes. It exceeds their optimal growth temperature, and it will denature the proteins and kill the bacteria. Fever also activates and increases the rate of other body's defenses, such as inflammation. The human inflammatory response is activated when tissues are damaged. In this example, there has been a surface wound. The first figure on the left shows normal, healthy skin. The white blood cells and red blood cells are freely flowing through the blood capillary, pictured here, enclosed within the epithelial tissue of the blood vessel. And so this is the blood vessel, and the white blood cells and the red blood cells are flowing through this blood capillary. Then there is a white blood cell here in the tissues that is just outside of that blood vessel. When the skin is punctured, bacteria and debris enter the wound, as shown here. The white blood cell binds to the bacteria and phagocytizes and digests that bacterial cell. The white blood cell now sends messages called cytokines to the rest of the immune system to notify them that there is a problem with foreign invaders. They tell them that they need to come right away and help to clear out this infection or clear out this debris that has enters the body through the break in the skin. These cytokines or signals trigger vasodilation, where the blood vessels dilate. So you see here between this figure and the next that the blood vessel has dilated. It's widened, and by dilating these blood vessels, it allows more blood carrying more white blood cells to get to that wounded area of the skin. So these blood vessels dilate, they expand in width, and they also become leaky to allow diapedesis. So diapedesis is the movement of white blood cells out of the blood vessels and into the tissues. So these white blood cells that are normally flowing through the blood capillary can now leave the blood vessel, squeeze through the epithelial cells of the blood vessel wall into the tissue so that they can help clear up the bacteria and debris that has entered the wound. Fluid and proteins also leave the blood vessels and build up at the damaged site and cause heat, swelling, and redness. The extra fluid presses on nerves in the area and cause pain. You should be able to explain what causes these signs of inflammation, the redness, the swelling, and the pain, as well as the heat. The purpose of inflammation is to contain the infection so the bacteria don't enter the blood vessels and spread through the body, to localize the infection in response to that area, and the proteins and immune response and clotting help to restore tissue function. Interferon is a small protein made by certain white blood cells and tissue cells. The major function of interferon is to warn nearby cells of a viral infection. Recall that viruses are just sacs of protein with DNA or RNA inside. Viruses infect cells by binding to the outside of the host cell and injecting their DNA or RNA inside of it. The DNA then enters the nucleus and forces the cell to transcribe and translate viral proteins and more of the viral DNA or RNA. Once the new components of the viruses are made, the viral DNA or RNA is packaged inside of the proteins to make more virus particles. The new virus particles are then released from the cell, and they often go and infect other cells. The interferon protein is made by the infected cells and released. We can see that it's released and it is sent to nearby cells. So it binds the receptors on the surface of these uninfected nearby host cells and tells them that there is a virus going around. Those uninfected cells turn on genes to synthesize antiviral proteins. So when the virus tries to attack them, the antiviral protein degrades the viral DNA or RNA to stop virus production. Phagocytosis is another defense mechanism of the innate immune response. You should be very familiar with this by now. Phagocytic white blood cells like neutrophils, macrophages, dendritic cells, and B lymphocytes all have phagocytic capabilities. Recall in phagocytosis, the phagocyte is attracted to the bacterial cell and attaches to it. The phagocyte then engulfs the bacterium by forming a phagosome around it as it's engulfed. A lysosome that contains digestive enzymes fuses with the phagosome to kill and degrade the bacterial cell. If the foreign invader gets past the first and second lines of defense of the innate immune response, they are faced with the forces of the third line of defense, the cell-mediated response and the humoral response. The major players of the third line of defense are the T lymphocytes and the B lymphocytes, or the T cells and the B cells. B cells and T cells, along with other blood cells, are made in the bone marrow. B cells remain in the bone marrow to undergo a maturation stage, and T cells are sent to the thymus to mature. Once mature, they migrate to the secondary lymphoid tissues, like the lymph nodes and the spleen. The lymphatic system runs alongside of the vessels of our circulatory system. Its purpose is to filter and return fluid lost from the circulatory system back to the circulatory system. During the day, when we're up walking around and living our lives, fluid leaks from blood vessels into our tissues, which is why some of us have swelling in our lower legs if we're on our feet too much. The vessels of the lymphatic system take up that fluid from the tissues and filter it in the lymph nodes, where the B and T cells are stored and waiting to eat the junk that's being filtered out of the lymph fluid. The filtered lymph fluid is then returned to the blood vessels and returned to the circulatory system. Recall that the job of the immune system is to surveillance the area, recognize foreign invaders, and then get rid of them. Antigens that are phagocytized are processed, meaning they are engulfed and then digested. Then pieces of the digested cell are held out onto the surface of the macrophage by the MHC class I or MHC class II protein molecules. We've all seen kids chew up something and then stick out their tongue with pieces of food on it. If I draw a macrophage, imagine this is a macrophage, and this macrophage has engulfed a bacterial cell, And now it is going to use the MHC molecule to hold a little piece of that bacterial cell on the outside of the cell. So going back to our food and little kid example, the MHC molecule is like the tongue of the little kid, and the piece of digested cell is like the food being held out on the outside of that cell. The act of showing the food or pieces of antigen is called antigen presentation, and the cells that do this are antigen presenting cells. Both B and T cells have receptors on the surface of their cells. B cell receptors can bind free antigen, that is, antigens that are freely floating through the body, like bacterial cells. T cell receptors bind antigen that is attached to MHC molecules that are on antigen-presenting cells, like the macrophages, dendritic cells, neutrophils, and B cells. The job of the receptors on the B and T cells are to recognize and attach to non-self, also known as foreign molecules, recognize and not destroy our own host cells, and also to receive and transmit chemical messages to other immune cells so that they can also help get the infection under control. On these normal nucleated cells, we see MHC class I proteins on their surface. This tells any white blood cell that is surveillancing the body that this is a normal host cell and that they should just go ahead and leave it alone. Antigens or other foreign substances either don't have MHC molecules, as in the case with bacterial cells and viruses, or they are human cells that have been transplanted. Those types of cells have MHC proteins, but since they are not of that host and the MHC proteins may not be very similar to the person who's receiving the transplant, then the body will recognize it as an antigen and destroy those cells. So this is what happens when the wrong blood type is given or in transplant rejection. One human is given blood or tissue from another human, and because the MHC molecules are very different, the body views those transplanted tissue cells or the blood as foreign, and the immune system responds to get rid of those cells. So that's why these proteins are named MHC. MHC stands for Major Histocompatibility Class I or Class II proteins, because they are used to determine compatibility of blood and tissue between donors and recipients. Antigens can be presented by infected cells because all cells that have a nucleus have MHC class I proteins on their surface, like the cell on the left. So this infected cell can present some of the antigen on the surface of its cell by the MHC class I molecule. In addition, nucleated cells that are also phagocytic, like the macrophages, neutrophils, B cells, and dendritic cells, also have MHC class II molecules on the surface of their cells. You can see in this figure the MHC molecules as they present a little piece of the antigen. Antigens must meet certain criteria in order to activate the immune system to respond to it. The criteria are based on varying degrees of foreignness. The more different the antigen is from the host, the greater the response. Size also plays a role. The antigen must be large enough to be recognized by the immune system. For example, larger molecules are generally more antigenic. The shape of the antigen must fit into the receptor of the T cell or the antibody. An antigen has multiple epitopes or parts of the antigen that can be recognized by the immune system. If these parts are not accessible by the T cell receptor or antibody, then it will not trigger an immune response. Antibodies are proteins that are made by B lymphocytes and released into the circulatory system. They're made up of 4 polypeptides or protein chains. The 2 heavy chains, which are these 2 longer chains, and then the 2 light chains or the shorter chains. They bind antigens in order to tag them, kind of like sticky notes. When the antibody encounters the pathogen that triggered an immune response, it attaches to the organism by the variable regions that are shown here. So say, for example, there is a bacterial cell. We'll say bacterial cell. And then, by definition of an epitope, different parts of this bacterial cell can be recognized by receptors or by antigens. So each of these areas that I circled on the bacterium could possibly be considered an epitope, or just a little spot on the bacteria that can trigger an immune response. So the antibodies, let me erase one of these arrows. So the antibodies, they can bind to the epitope by the variable region. So each of these antibodies can bind to different antigens or 2 different epitopes, because there's 2 areas on that antibody. So the variable region binds to the epitope and tags that bacterial cell. Once it binds, it tags it so that the immune system knows that it is foreign and it needs to be destroyed. So then what happens is that a macrophage comes along, and the macrophage binds this region, which is called the constant region. So the large macrophage, I'm running out of room here, this is the macrophage. It binds to the constant region of the antibody, and then it engulfs this entire bacterium along with the antibody in order to phagocytize it and destroy it. There are different types of T cells and B cells, and you should be familiar with them all and the roles they play in their immune response. The adaptive immune response is divided into the cell-mediated response and the humoral response. At the center of both types of responses is the T helper cells that activate T cytotoxic cells and macrophages of the cell-mediated response, and also activate the B cells involved in the humoral response. The T helper cell is activated by an antigen-presenting cell that is presenting a piece of antigen on its MHC class II molecule. And then the T helper cell activates the cell-mediated immune response and humoral immune response based on the type of antigen that was presented to it. T cytotoxic cells destroy host cells like those that have lost their MHC molecule, for example, the cancer cells or the tumor cells, virus-infected cells, and also transplant cells. B cells are involved in humoral immunity. Humor is another name for blood. And B cells fight antigens by releasing antibodies into the blood. B cells can also be activated by T helper cells, as well as bind free antigen. Once they become activated, they differentiate into plasma cells and memory cells. The plasma cells make a bunch of antibodies and release them into the blood to circulate through the body and bind antigen so they can be phagocytized later. Memory B cells hang around in the body for a while after the infection. If the body is infected with the same antigen with the same epitope, this will trigger the memory cells to differentiate right away into plasma cells and then pump out antibodies to help get rid of the infection. Let's take a look at the cell-mediated response. It is called the cell-mediated response because the T cytotoxic cells or the phagocytic cells are actually doing the work by binding the antigen directly, versus in the humoral response when the B cells make antibodies that bind the antigen. So in the cell-mediated response, T helper cells have been activated by binding to an antigen on an MHC class II molecule of an antigen-presenting cell. So this figure does not show that antigen-presenting cell, but let's just draw this. Let's draw an antigen-presenting cell. We'll draw the MHC class II molecule, and then we will put a little antigen on the MHC class II molecule. So this antigen-presenting cell presents this antigen to the T helper cell, and then this T helper cell becomes activated. The T helper cell can then activate that macrophage that is presenting that MHC class molecule, and once that macrophage becomes activated, it then triggers the macrophage to digest the bacterial cell or whatever it has engulfed. It activates it to digest it. And so the macrophage will then destroy the contents inside of it using the digestive enzymes of the lysosome. T helper cells also send signals to the T cytotoxic cells. These T cytotoxic cells are going to destroy host cells that have lost their MHC class I molecule, and they also destroy those virus-infected cells. On the flip side, the T helper cell that has been presented with antigen can also activate the B cell or the humoral immune response. Once activated, the B cells differentiate into a plasma cell and a memory cell. The plasma cell enlarges and makes a bunch, just a whole bunch of antibodies that are specific for the original antigen that activated the T helper cell. Those antibodies are sent out into the blood circulation to bind free antigen. So this figure shows you the antibodies binding to the antigen. Now this antigen is tagged for destruction by phagocytes. The phagocytes or the macrophages then come and they bind to this region of the antibody, and then they will engulf that entire organism along with the antibodies and phagocytize it. Recall that the B cells can also bind free antigen via their B cell receptors, which is happening here in the beginning. So this B cell has bound this free antigen. When they bind free antigen, they can also present it to the T helper cell. When they present to the T helper cell, then the T helper cell activates the immune response. This is an overview of both the humoral and cell-mediated responses. Note that both T and B cells form memory cells. The presence of these memory cells allow the immune system to respond faster and stronger when infected with the same antigen. This concept of exposing the human body to antigens to trigger a response in memory cells is basically the basis for vaccination. Understanding how the natural immune system works helps us recognize problems and guide treatment in those with immune disorders or to boost our own immune systems. Natural immunity is acquired as part of normal life experiences. And artificial immunity is acquired through a medical procedure, such as giving a vaccine or actually giving someone antibodies. Both types of immunity can be active or passive. Active means when the body makes its own antibodies, it's naturally and actively making its own antibodies in response to encountering a pathogen. Passive immunity is when antibodies are given, either they're given naturally or they're given in a medical setting. In these figures here, we see that this person, this young boy, is sick. So he has encountered an antigen through his normal life experiences, and his body is actively making antibodies against the antigen that he encountered. Let's compare that to artificial active immunity. So this baby is being introduced artificially to an antigen by injecting it as a vaccination. But even though this baby is not encountering the antigen naturally, it's encountering it artificially, it's still actively making antibodies against that antigen. Now let's talk about natural passive immunity. So passive immunity, again, is when antibodies are given. They can be given naturally through the process of breastfeeding. Through breastfeeding, the mother is passing antibodies that have been made or preformed in her body. She's passing them through her breast milk to the baby. In artificial passive immunity, now let's compare this to artificial passive immunity, where this little girl is having a problem making her own antibodies. So instead of making her body being able to make antibodies or receiving them from someone else, she can receive antibodies through an IV in a medical setting. So in both cases of natural passive immunity and artificial passive immunity, here antibodies are given to the baby through the breast milk, and here select antibodies are given to someone who cannot make their antibodies. So be able to explain the differences between these different types of immunity. With active immunity, whether it's natural active immunity or artificial active immunity, these people are responding to an antigen. So their immune system is responding to the antigen, going through the immune system or the immune response, and then the result is going to be memory cells. But with passive immunity, that person is being given antibodies. And so the antibodies will not trigger an immune response. We're kind of just giving them the final product. So their body has not encountered an antigen and gone through the immune response to make memory cells. So those antibodies will only last in their system to help fight that infection for a short period of time until those antibodies are given again. So in people that can make their own antibodies, eventually they will encounter those antigens and make their own antibodies. But in the people who have some type of genetic disorder where they can't make those antibodies, they have to be given antibodies on a regular basis. As mentioned, vaccination is a type of artificial active immunity. A person is deliberately exposed to material that is antigenic but not pathogenic. That's the difference. It will elicit an immune response, but it will not cause infection. So this person is exposed to material that can trigger an immune response, but it will not cause infection. And so that's just the purpose of a vaccine, is to trigger an immune response so that our bodies can make memory cells and antibodies. After the first exposure to that antigen, it takes a while for the body to respond and begin to produce antibodies. So we see on this chart here that the levels of antibody after the primary immune response, it takes a while for them to actually start making for the body to start making the antibodies. And then compared to the second response, this is only the level of antibodies that are made. After the second exposure, we see that the time that it starts to make between the exposure time, which is here, and the time that it takes to really start making lots of antibodies is a lot shorter than it is the first time. So the response is both faster, and the antibody levels are much, much higher than they are after the second exposure. And this is basically that second exposure, when the response is both faster and stronger, is pretty much due to those memory cells. So these memory B cells and the memory T cells that are hanging around in the body after the infection is over.